Okay, good morning everyone. Welcome to the RACI New South Wales Pharmaceutical Sciences Group webinar. Today's webinar is on vaccine design and manufacture. My name's Nisha Davis and I'll be your host today. It's my absolute pleasure to be part of this program. This webinar is third in a series of webinars the RACI Pharmaceutical Sciences Group are presenting on the aspects of the science of COVID-19. Today, we're talking about the question that the rest of the world is talking about with regards to the race to design and deliver a vaccine for COVID-19. Our presenters today are in this race and we're very excited to hear what they have to say. First of all, I'd like to talk about the format of today's presentation, which will be a 20-minute talk by each presenter, followed by a Q&A. We'll try our best to get through as many questions as possible, as I'm sure there will be lots, but it will be time permitting. This webinar will also be recorded as well and available on our website for future review. To ask a question, please use the Q&A function. And all your questions will be anonymous as well. Today's speakers, it's a pleasure to introduce them both. Speaker one is Professor Trent Munro, and he'll be talking about developing vaccines at speed. He's the Senior Group Leader at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland and Director of the NCRIS-funded National Biologics Facility. His research is focused on the development, engineering and production of biologics using mammalian cell culture systems. Professor Munro is also a Program Director of the CEPI-funded Rapid Response Vaccine Pipeline, and this role has been part of the University of Queensland team developing a clinical stage vaccine candidate for COVID-19. Dr. Paul Howley is the Managing Director of Vaxmed. Vaxmed is developing a new vaccine platform technology with a new standardized procedures for vaccine design, construction and manufacturing. Synvax vaccines have the potency of attenuated vaccines, the safety of inactivated vaccines and the versatility of subunit vaccines. Dr. Howley is the inventor of Vaxmed's Synvax platform vaccine technology and the vaccines in development. He has decades of experience managing vaccine development programs and expertise in the areas of antigen design and discovery, proof of concept studies in animal models, GLP preclinical and toxicology studies, process development, GMP manufacturing, regulatory affairs, and first-in-man studies concerning live viral vectored vaccines. So our two speakers today are very much in this race and the eyes of the world are very much watching the success of these programs. And first of all, I'd like to hand over to our first speaker, Professor Trent Munro.
Thanks very much, Nisha. Nice to be here. I'll just pull up my slides, see if this works. Okay, so hopefully that all good, Nisha, from your side?
That looks great.
Great. All right. Yeah, thanks everyone. Yeah, pleasure to be here, as I said, and have a chance to talk about the program that we've been working on here at University of Queensland. Big team, both here and across Australia. So a shout out to our partners, Australian partners at CSIRO, Doherty and ANU. And I'll try and credit as many people as I can as we go through, and I've listed the rest of the team there. Okay, so I mean, if we step back, I think to the end of 2019, you know, the focus for Australia pretty much was on what was happening from the bushfire crisis, and I think we all expected that that was going to be the recovery event for 2020. And I know that all of my international friends were calling and asking about how things were going. Little did we know this wasn't going to be the worst of what we were going to see globally in 2020. If we flick backwards a little bit, you know, I think if you'd ask people what pandemics are about, you know, various images probably come to mind, but probably one of the most well known was that, since, around 1976, there'd been ongoing Ebola outbreaks in Africa, and the vision was always like this. It was distant, it was kind of in our minds, we knew it was happening, there was a general fear of this type of disease, but it was also sort of out of view, and aside from, the odd patient popping up in Western countries or repatriated to Western countries, this wasn't something that really was a focus for many of us. But, I think emerging pandemics, especially zoonotic-based disease, had been in the minds of lots of people. And, whether they'd been shouting into the wind or not, the idea of having a preparedness strategy across the globe was something that really came into focus, I think, after the 2014, 15, 16 Ebola outbreak. And one of the things that thankfully came out of that, because of the woeful international response, was the foundation of this organization that we call CEPI, but it's a bit of a mouthful. It's the Coalition for Epidemic Preparedness Innovations, and that organization was essentially founded and funded to the tune of a couple of $2 billion to invest in technologies that could help us bring technologies that would be used as vaccines for emerging pathogens. So, thankfully, this organization did exist. It's been one of the catalysts, I think, in this COVID response, amongst many others, but, had made some pre-existing investments, which I'll talk to you about. So the way that CEPI was set up was that they initially looked at some priority pathogens that you may have heard of, other diseases which could represent significant global health threats, things like MERS, Lassa, Nipah, chikungunya, Rift Valley fever, and they had some others. And they also had this interest in something they called disease X, which was essentially, what would happen if we had an unknown disease then cause a global event. So, thoughtful process to at least have someone with quite a lot of cash, at least for an NGO, to invest in this whole area. And one of the things that they did back in 2018 was to put out a call for people who had platforms that might be applicable for rapid vaccine development for epidemic-prone infections. And so, very aggressive type of program, sort of blue sky thinking, a bit like what you might see in a BARDA type program. 16 weeks from identification to clinical trial material, a 6-week time frame from first dose to clinical benefit, and then something like 100,000 doses within 8 weeks. And these were the aspirational targets for this whole platform call that went out. So I'm going to come back and talk about why this is important in a bit. But first of all, I need to tell you a little bit about vaccine development and a particular type of vaccine. And those are vaccines that are based on proteins that are found on the surface of viruses. So if you have a look here, it's an exemplar viral particle. You can see these red things here are basically the surface protein on the virus, and they're typically what the immune system will encounter. Importantly, they're also what is used essentially to dock and target and infect the target cells of the body that the virus will be attracted to. And one of the really interesting things about these viruses is that while there's a number of classes, and this whole recognition fusion event is very, very complicated, one of the primary ways this happens, especially in type 1 viruses, is that when the protein is usually on the surface of the virus, it's held in this pre-fusion conformation. Upon docking or upon binding with a receptor, it flips to this post-fusion conformation. And if you try and make these proteins in isolation, quite often what you're making is this post-fusion form, which is lacking accessibility to the things that the immune system really needs to try and access if you were to have an effective immune response. So that's really been the big challenge for subunit vaccine development, and is something that the University of Queensland group here had been working on for the last 10 years. And so the technology that they came up with, and this is out of Professor Paul Young's lab, and two of the inventors, Associate Professor Keith Chappell and Dr. Dan Waterson, who came up with this whole technology. So basically to use a clamp here and to attach this clamp, to append it to these viral proteins so that you could try and lock this trimeric structure here, which is the surface molecule on the virus, into this pre-fusion conformation. And there are some other technologies out there that have attempted to do this in the past, and some are being used for COVID vaccines now. There's several potential advantages to this molecular clamp format. One is the particular clamp that's used has a really high degree of thermal stability, and we're hoping that that makes it a differentiator. And also that it's a very strong trimerization motif, which means you form a very stable trimer and over time, and that we hope that the combination of that stable trimer with being able to at least lock a proportion of this molecule into the pre-fusion conformation will hopefully lead to an effective vaccine. So the team had been working on this, as I said, for 10 years basically, and they looked at the applicability of adding this clamp to a whole range of different viral proteins, so things like RSV, flu, Ebola, MERS, Lassa, Nipah. And so they had this body of data to demonstrate that this, from a proof of concept point of view, looked really promising. And so what they did was they decided they put a proposal in to this call that I told you about from CEPI, this platform technology call for funding. And they'd been struggling to get funding for quite a number of years from traditional approaches. And so this CEPI opportunity was really a nice fit. And one of the things they did was come up with this rapid response vaccine pipeline. Essentially, it was basically saying that we think because of some of the advantages of the platform, some I won't talk about and some I will, we think we can go really fast from pathogen identification through to having some clinical material. And so they did that, and they were actually successful in that call. And it was a very significant amount of money, at least for platform validation, about 15 million Australian dollars, that was then given to the group to say, look, okay, we think your platform has promise. You put together a program. Let's see if we can get some clinical proof of concept. And so that happened back in early 2019. So the team was given that money, and they finally had some of the resources to try and validate this platform. And the plan they had was essentially to look at influenza first, go from discovery to clinic, look at MERS, discovery to clinic, do some additional optimization work on RSV. And then at the end of that, for the last part of the grant, was basically to take all of the learnings from these other programs and do a mock emergency response. And so the idea would be to go from DNA to dose as quickly as possible. And the idea was that we would have had a couple of years to understand all of these aspects and have clinical data, a lot of preclinical data, and be ready to roll on disease X. So that was the plan that happened. They wanted someone with some industry experience on the program. I came back to Australia from working in industry in the US last year. And so I joined the team about August 2019. I've got a few roles here at University of Queensland, and one of them, as was mentioned in the introduction, was to take on the role of director for this CEPI program. So, of course, as it turns out, as you all know, we didn't have the 2 years to prepare to really understand our platform and to do everything we wanted to do, because late last year, very late last year, WHO surveillance picked up this cluster of pneumonia of unknown cause in China. And there was quite a number of people at the time who were saying, well, we really think this could be disease X. I'm not sure how many people really believe that in December or even early January. But of course, we now know a lot more about it. And that was this SARS-CoV-2 virus, which causes the disease Covid19. And so with that, the view of pandemics quickly changed, right? Our view of a pandemic affecting people, far away in Africa, changed to images like this, where, all of a sudden in Western developed countries around the world, you're having the best healthcare systems brought to their knees by an influx of patients and an inability to do anything for those with severe disease. So very, very different type of situation from that I showed you earlier. Of course, don't need to spend any time on this. Everyone, I'm sure, is following various trackers. Over 15 million cases now, I think, well over 500,000 deaths, and it won't be long before we hit 1 million. So a very, very severe disease. And you can see in this nice visualization here, the ebb and the flow through all of the different major continents and countries This is truly a global health catastrophe that we're facing. Okay, so we get to vaccine. How fast can we go? This has been a stress test globally for lots of different things, and will we be successful? So, I think back in January, February, people were saying that to develop a vaccine would take around 10 years, typically 5 to 10 years, maybe longer, maybe 20 years. And then, as we were thinking about how fast we could go towards having broad distribution of a COVID vaccine, I think conservatively, people were saying, well, maybe it's 12 months, maybe it's 18 months, maybe it's 2 years. That timeline, seemed incredibly ambitious and incredibly fast. But as we're quickly finding out, it's probably not going to be fast enough. We don't know if we're going to hit those timelines. But I can definitely say that every major milestone that has come up, the scientific community has hit it faster than we were thinking. So I think we're optimistic, but it's still early days. All right, so quickly to run through what we've done in our UQ program, and then I'll come back and tell you where I think the sort of global situation is for vaccine development. So for us, back on January 23rd, CEPI asked us to pivot all of our resources on our rapid response platform and take everything that we knew and throw it at COVID-19. So we stopped all of our other CEPI programs and focused just on COVID. Fortunately, the CLAMP approach was a very good technology fit for this particular virus. We had meetings back in very early January with CEPI, where it was clear that people were concerned. And as soon as the DNA sequence came out, the team hit the ground running. And we were already well into thinking about how we were going to do this by the time we signed this agreement in that week of January. It was a rush of activities over the next few weeks, from signing various agreements to get access to commercial adjuvants. So we had commercial adjuvants from Dynavax and GSK, and also Securus, a CSL company. And then, some sleepless nights as we tried to find a lead candidate. And really, this is where the team was really pulling the long days in the lab, an incredible amount of work from the team here to come up with our first initial candidate that we thought had promise. We locked that in back on Valentine's Day. And, at every step, you can see the smiles on people's faces as data was literally rolling off. And we were able to get some nice EM images of what we thought was going to be our final candidate vaccine, and things were looking pretty good. So that was back in February. We took a small amount of time to have a quick celebration. I think it was a lot of relief that day. Someone made a Corona Trimer. I'm not sure if that's appropriate, but it was quite humorous back then for us. And this was, I think, still pre-social distancing, when no one was really sure about human transmission dynamics at that point back in February. We then quickly realized that if we looked at our initial cartoon here, it was of course overly simplified, and that we were going to need to do something really dramatic if we were going to need to push forward. While we had some really good financial support from CEPI, we were talking to a whole range of commercial providers, and we knew that we were going to have to spend a lot more money. And so one of the things we did very early on was basically talk to people in government, people in industry, people from philanthropic sources, to realize that we were going to need a real cash injection if we were going to work in an unconstrained environment. So, and the reason for that is that was going to drag all of our activities right back here before Q2 with this funding, to basically head into some deep process development and then straight into large-scale manufacturing in parallel with all our preclinical and clinical studies. So that was a huge amount of work. We flew around the world at that time, when you could still fly. And thankfully, we were able to do that. And in fact, we initially announced a $17 million injection of funds, and just subsequently it's up around $25 million. And that really meant that we could work with a whole bunch of different companies. We could start paying for all the contracts we needed. We could pay for all of the work here. We could, do all of the animal work we needed internationally, which cost millions and millions of dollars. And some of the manufacturing aspects of this, we were signing $2-3 million contracts at a fairly regular clip. So all of that meant that we could move at speed and not really worry about the financial side of it, at least for this very early development work. Of course, to develop a vaccine costs hundreds of millions, if not billions. But this initial work, at least, is what's let us get into our proof of concept. We then had some good animal data in April. We were able to show that we saw some really promising immunogenicity data. And then we moved into manufacturing. And so there was two parallel things going on. One is we were working both here at our facility in Queensland and with our colleagues down at CSIRO in Clayton to do manufacturing for the clinical trial. So that was very, very intense period, moving out of the lab and into the R&D, out of the R&D lab and into the pilot scale labs where we have bioprocess capabilities. We're able to produce our preclinical and clinical batches. And at the same time, we formed a strategic partnership with CSL, who've agreed to be the manufacturer of choice for us. Should the data roll out the way we think it hopefully will, they'll manufacture and take over clinical beyond our phase 1 study, which I'll get to. So that happened on June 5th, although we'd been working with CSL way back. In fact, the first meeting we had with CSL, I think, was back in January. So very, very good partnership with them. And we'd been receiving ongoing support from other companies as well, like GSK and others. So really great collaborative effort to get to that point. But, clearly as a university group, we knew we needed a large manufacturer to come on board if we were going to make this happen. We then have been doing key animal protection data. So this is basically looking at animal-based models of efficacy. We've been doing that over in the Netherlands. At the time of getting this material over to the Netherlands, basically there was no planes flying. I think, we had weekends where we were talking to every shipping company in the world to try and get material over there to start these studies. So very, very challenging environment, but we did, and that work's ongoing. We had some initial data back, looks good. And then, of course, just a week or so ago, we hit our most important milestone, and that was our first human dose, which happened on July 13. And I think, we'd said from January that our goal was to be in the clinic in mid-year. We knew that this being a protein-based vaccine, and because of the nature of the manufacturing, we weren't going to be able to move as fast as, say, the nucleic acid-based or viral vector-based vaccines. And we were really very, very happy that we were able to stay on track. And I think our first date for clinic was actually June 29th. So to actually hit June 13th, we considered that a huge win. So that was great. That started. We're through one cohort, about to start another cohort. So there'll be about 120 patients who will go through that clinical trial, and we're hoping to have our initial readout from that, an interim readout within the next couple of months, and then we can compare it to all the other clinical data that's quickly coming out from the other platforms that some of you may have seen. So, for us, it's been an incredibly crazy period, 5 months from lead candidate to human dosing. Sort of really, hit a lot of the things that we thought we were going to do, and we're very thankful to the broad support we've had both within Australia and internationally for this program. Okay, just to finish then, I think I'm nearly out of time, we're running out of time. If we look at vaccine response in previous sort of pandemic situations, SARS took, I think, around 20 months to get from DNA available into a clinical study. If we look at H5N1 flu, it was about 11 months. For H1N1, it was 4 months, but you have to remember with influenza, these are known platforms. This is something that we do all the time. For Zika, there was some really interesting efforts that got into clinic very, very quickly. And then I think remarkably, for the current situation, Moderna was able to get from DNA sequence to dosing their first patient in just, I think it was 65, 66 days. So an incredible effort in collaboration with the NIH for their RNA-based platform. And they're well on their way, as you may have seen in the news, to they've presented some of their phase 1 data and are moving into later stage trials. So in terms of how fast science can go, everything is being blown out of the water with this pandemic. And, I think that, hopefully we'll be able to bring forward an effective vaccine. So if you want to know what's going on globally, here's just a little snapshot from New York Times. It's got a great vaccine tracker if you want to look it up. There's a whole range of different platforms, and we can talk more in the Q&A if anyone wants to know about these different platforms, from genetic vaccines to viral vectors to proteins to whole virus. Obviously, we're in the protein vaccine bucket here, and you can just see by the number of different companies that are engaged here. This is a huge global effort. And just to say, to finish with from that same vaccine tracker of, where things are at, there's more than 140 in preclinical, probably more than 200 actually. There's 19 programs in phase 1, including us. Already 13 programs which have done a combined phase 1, phase 2. So that really depends how you sort of classify your trial. But these are additive numbers, so unbelievable. And now there's 4 companies that are really on the cusp of starting very large phase 3 efficacy studies. So just to be a few months and to be very, very early in this whole pandemic, and to already have a number of vaccine platforms entering large-scale pivotal trials is kind of amazing. I think it's far ahead of where we thought we'd be. We've got a lot to learn. It's super early days, but people are feeling somewhat optimistic. These coronaviruses are definitely challenging. We know a lot about them, especially from the veterinary field. We know a lot about them from common cold research, and we should know a lot more from what happened with SARS and MERS. But of course, as people will understand, because those diseases faded out of view so quickly, a lot of the research on those programs unfortunately didn't go ahead. And if it had have kept going, we may have even been much further ahead. And of course, there's one here sitting in the approval bucket, and this is out of China, where it's actually being given, as far as the news reports say, to the Chinese military as an approved actual vaccine. So, we'll see. Everyone's watching. We can talk more about all of the caveats to this if people have questions, some of the things that people are watching out for from the vaccine development side of things. But incredible scientific endeavor over a few short months. So with that, I'm going to finish. Obviously, we didn't do this alone. So a huge number of people have leaned in to help us, either providing technology, providing money, providing facilities, providing expertise. So we're very, very grateful for all of that. And thank you for listening and giving me a chance to talk about our program.
Thank you, Trent. That presentation was amazing. I feel that we're in good hands with you and your team in developing this vaccine. That's much needed. We do have a question from the group, from the attendees, if I can sneak it in before our next presentation. It's around, the term effective. What makes a good vaccine? Is it quite possible that some of these vaccines that are coming eventually to market will bring equally successful outcomes, or is it likely they will differ in their success?
Yeah, I think you kind of summed it up, and I think they will be different. I think we'll learn quickly. I think the one thing that sort of seems to be becoming apparent, that it seems unlikely at the moment that this will be a one-shot type of vaccine approach where you'll get long-lasting immunity. It's highly likely that you'll need a primary shot, a booster shot, and you may even need, some type of booster at some type of cadence after that. That seems, even if we're successful, that seems most likely. It may even be that we need to combine different types of vaccines. So we could take a prime boost based strategy where, say, you take a nucleic acid or a viral vector as your prime, as the initial vaccine you get, and maybe you come in afterwards with something like an adjuvanted protein vaccine like ours, and we'll see how that plays out over the medium term.
Great, thank you again for your talk, and we'll have more questions after Paul's presentation. So I'd like to hand over to Paul.
Thank you, Nisha. Let me just call up my slides. Oops. 1 second. Great. So I've been asked to give a talk, and a colleague of mine have actually proposed a title for me to talk about formulation, upscale, and manufacturing of vaccines. I'm a molecular biologist. I was told not to really make it too much biology, as this is a chemistry society thing. So, regarding formulation, I'll just talk about what our vaccine is, the active ingredient. We're, as a company, more interested in the manufacturing and upscaling side of this thing, because I think that's the important thing right now that's needed is manufacturing capabilities at scale. So Vaxmed is an Australian biotechnology company, and what we do is design and develop vaccines for unmet needs. It's a new company. We just started a couple of months ago, really to start a new, basically develop a new technology for making pox-viral vector vaccines. Our technology enables us to make safe and effective vaccines, and again, with the precedence of speed and scalability. With our technology, we can standardize the way we make vaccines and manufacture them. And really, with this system, we believe that it has some of the advantages of being a live attenuated vaccine like the measles, mumps, rubella, but has the safety of inactivated vaccines, and then the versatility of subunit vaccines. So it's really all those three advantages rolled up into one. Reason why we've chosen the pox viruses is that they're quite potent in stimulating immune responses. They're very stable. The DNA virus, they can be easily genetically engineered, and if you can actually create, you can actually standardize the whole process for making a number of vaccines in what you call an assembly line process. We don't have the money to take these vaccines all the way to commercialization, so our business model is really to partner these off with the big pharma companies that are interested. So our mission really is to use these vector systems to streamline the process. Because you're using a common sort of delivery system, the vector, when you come to do things like toxicology, it starts to become standardized, because the main active ingredient is actually your vector. Your vaccine is your vector, which carries your genes for antigens, and your antigens are normally derived from, I guess, like surface proteins, structural proteins, which sort of benign in nature. They don't have any biological functions, not enzymes or mediate certain chemical reactions or anything like that. They're just there for the immune system to see. So in doing that, in generating, I guess, a wealth of information about your vector system, then when you come to do things like toxicology, once you've done it a few times and you're showing that the vector system is safe, then when you come, as in this case, in emergency, you can quickly make a vaccine without having to worry about spending 6 months doing a toxicology study. And that's a great time-saving thing. We aim to really just standardize the whole process so it becomes fully validated, rapid scale up, and keep the cost of goods for manufacturing really low. They're the things that we're trying to do. And if we can achieve this by keeping everything consistent, and also looking at, a dosing, we know from background experience that, this is a matter of the dose you need to stimulate the desired immune response. We're looking at the certain types of immune response we want to see for diseases. We're big into a broad immune response, not just antibodies or neutralizing antibodies. There are antibodies that are non-neutralizing that play a role in clearing viral infections. There's cell-mediated immune responses. These are where like immune cells come along and kill infected cells to stop the spread of infection. So these are the things that we want to make sure that, you know, with our vector system, we can create them in a consistent manner. So in times of emergency like this, then we know that, you know, by putting, I guess, the antigens from whatever disease is out there, that we've got a good chance that this vaccine will be appropriate for controlling that disease. So it's really just, you know, a consistent way of designing the antigens going there, the construction of the vaccine. We want to do it as quickly as possible, use a universal manufacturing process that we can scale up or down at will. And we got a clear pathway into clinical trials, and we know how to design the clinical trials. So that in going further, when you're looking at efficacy, then that's the variable that otherwise everything's just the same. And to coin a term that one of my business partners is telling everybody, it's all about just-in-time vaccines, and also creating, I guess, a vaccine vault by making these vaccine constructs for all sort of diseases, putting them away in storage until a time is needed that we can then bring them out and use them for, you know, for epidemic or pandemic purposes. And one of the things that we're sort of looking at is influenza, because that keeps coming up every now and again. So having something put aside there, ready and waiting, that's going to be a big help. So, as I mentioned before, our vector of choice is vaccinia virus. It's a non-replicating vaccinia virus. I'm a pox virologist, but I have worked with adenoviruses and some of the RNA viruses, and I've done some subunit vaccines, and I just find that they're the easiest to use, and they have a lot of advantages that the other viruses don't. And one of the big advantages I like about pox viruses is that they can accommodate a large amount of genetic information. So you can encode many different antigens from the same disease virus, or you can create a single vector multivalent vaccine. And one of the projects I was previously been working on was developing a chikungunya and Zika virus single vector vaccine. And the reason for that was that the chikungunya and Zika viruses infections, they're like brothers and sisters. You know, they occur together. They're transmitted by the same mosquito vector. So it seems sort of logical to make a combined vaccine for those two diseases. Vaccinia virus was the smallpox vaccine. It was very effective at eradicating smallpox. In the 80s, 90s, it was also used as a vector vaccine for rabies. And it was successfully used in Europe to eradicate rabies in the northern parts of Europe by using fox bait laced with vaccine, which were dropped from helicopters. This is, for me, is quite a significant achievement, and it shows how, you know, how these pox virus vectors are really showing, you know, really good, in the sense that, you know, these things are also thermostable. Yeah? And the fact that you know, within 10 years they could eradicate rabies shows that, you know, these are really amenable viruses for creating vector vaccines. They're highly immunogenic, they're self-adjuvanting. You don't need adjuvants for these things. They promote a very strong potent immune response. And that immune response is a balanced one with antibodies and cell-mediated responses. It's a these are DNA genomic viruses, which means they're extremely stable, unlike the RNA viruses. So some of these RNA vector vaccines, they are prone to mutations and changes, and coronavirus is an RNA virus, and as it's going around the world, you notice that people have noticed that mutations occurring. Coronavirus is one of the RNA viruses that's less prone to mutations, but you look at something like HIV, they have a high rate of mutations. So the pox virus is physically robust as well, which is good for, I guess, manufacturing purposes. They can withstand high shear forces. They can be easily desiccated, and as I mentioned before, they're thermostable. So this makes them amenable for but developing very rigorous purification procedures where you can use sonication, high pressure modularization, centrifugation, column chromatography without losing too much of your virus. They don't need to be formulated. They're quite stable in a tris saline buffer. They can be easily freeze-dried without the need for protein stabilizers. Liquid formulation, they can be stable in the fridge for a couple of months, a couple of years at -20 and definitely at -70. And with freeze-dried formulations, they can last for a very long time at room temperature for years. The other thing I like about them, they do have a broad host range for cell substrates for manufacturing. They can be easily manufactured in MRC5 cells, which is the cell line used to produce the registered chickenpox vaccine, Viros, which is used for polio and Japanese encephalitis, WI38, which is used for rubella and adenovirus vaccines, HEC293s, they're used for manufacturing the vectored adenoviruses, the non-replicating ones. MDCK to grow quite well in those. These cell lines are now being used to produce the cell-based influenza vaccines, and the chicken embryo fibroblast cells, CEFs, which is used for measles, influenza, and rabies. So there's a broad range of cell substrates to use for manufacturing. So back in the during the smallpox vaccine era, and we're looking at the early 1800s and late 1700s, when there was no science as such as there is today for manufacturing. The only way they could produce this smallpox vaccine was actually from the pustules of on the side of infected cows. And if you see this picture here, this is a picture taken around about in the early 1900s, and you often hear of like the serum institutes. Well, those institutes were really born out from actually producing smallpox vaccine from animals. And the way they did this was that they would scratch a seed vaccine onto the side of the cow, wait for a blister and a pustule to develop, and then they would harvest the liquid from those pustules or the crust from the scabs, then homogenize it together and then filter it to remove all like hairs and insolubles. And then during the, I guess in the smallpox eradication program, when they learned how to do titrations in eggs, they would titer and then dilute to 10 to the 8 infectious units, freeze dried, and then reconstitute in a water glycerol solution. And the way that you would be vaccinated is using a bifurcated needle, and then you dip this needle into your vaccine vial, and then basically scratch it onto your skin, and that needle contains about 2 microliters. So you get a really low dose of vaccine compared to what people would be getting with MVA or with adenovirus. Now that vaccine was often referred to as calf lymph vaccine, and that method of manufacturing was done all the way up to 1980 when smallpox was declared eradicated. However, with I guess with 9/11 occurring and the need for maybe we should keep making this vaccine, because if there's rogue states that are going to be using this as a bioterrorism or biowarfare weapon, then we need to establish stocks. But obviously in today's era, you can't manufacture vaccines from the side of cows, especially when you have the likes of BSC and other diseases that are rampant in cattle that has to be screened for. So the second generation smallpox vaccine is actually a tissue culture produced vaccine, and the cell line there, the preferred cell line is Vera cells. And what you do is grow those cells into these roll bottles here, and that's just one rack, and you'd have hundreds of these racks going, and then you just inoculate them with your seed vaccine, wait for the cells to show signs of infection, harvest those cells. This pox viruses are not secreted out of the cells, they're actually within the cell and on the surface of the cell, so you have to extract that virus from the infected cell, clarify it, purified by centrifugation and then formulation in a saline buffer. And nowadays people use PBS instead of citrate buffer, tighter, and then dilute down to 10 to the 8 and freeze dry. And vaccination is very much the same as it was before, given by a bifurcated needle that's scratched onto the surface of your skin. And here's just a picture of ACAM 2000, which is manufactured by Cambis. And the Americans have stockpiled, I think, 300 million doses of this in preparation that if somebody does intentionally release smallpox, that they've got enough there to vaccinate the population. There's a problem with the smallpox vaccine. I mean, it did eradicate smallpox, but there were side effects, and a number of side effects. And these side effects occurred in 1 in 100,000 vaccinations. And this picture there is just one of them. And this is called progressive vaccinia, where at the inoculation site, the smallpox vaccine has actually spread and causing necrosis, and that will spread, as you can see, it's starting there. And that really can be only controlled by giving an antibody to vaccinia virus. The vaccination did result in death. About 1 in a million people died from smallpox vaccination. But if you compare with this with a death rate of 1 in 3 with actual smallpox infection, then the risk benefit is it's working towards the vaccine. However, now that smallpox is eradicated, the WHO said that these side effects, the rate of these side effects is no longer acceptable, and they're looking for 0 rather than 1 in a million or 1 in 100,000. This was during the eradication program, everybody was very aware of this, and a number of approaches to attenuate the smallpox vaccine had started there, and one of them is called MVA. Now, MVA is a smallpox vaccine that's been serially passaged in Ceph cells. Ceph cells are avian cells, and the way that most, vaccines are made is by attenuating the disease agent in a cell line that's distantly related to, the target population. As in humans, avians is as far away as you can get, so that virus then adapts to that species and no longer becomes virulent to the host species. For example, measles virus, was attenuated in Ceph cells, so it lost its pathogenicity to humans, but it still was able to replicate and be a very effective vaccine. However, when they attenuated smallpox vaccine, in the case of MVA, it did lose its potency. And because of that, you need multiple doses. You need to have a prime boost regime to create a strong, robust immune response. Now you've adapted to Ceph cells, it's lost its broad host range for a cell substrate for manufacturing, so it can only be produced in Ceph cells. Now, Ceph cells are chicken embryo fibroblast cells. They're prepared freshly from 12-day-old chicken embryos, and then those embryos are basically mashed up into soup and then cultured onto plates. So it's a bit of a gross process. And because you're relying on fertilized eggs and then you're incubating those for 10 days or 11 days, and then extracting the embryos and then the cells from those embryos and culturing, it can be quite a long process to produce a batch of vaccine, and you don't get batch to batch consistency. It's quite variable. And one of the drawbacks also is that these eggs have to be pathogen specific free. And they monitor that with each of the flocks, and the results are retrospective in the sense that you won't get from a batch of eggs, you won't get results from the testing until 6 months later. So if you've gone ahead and manufactured a couple of batches of vaccines and the results come back and say, oh yeah, that one of those birds in that flock was contaminated with a adventitious agent, then that whole batch has to basically go into the bin, and then that whole, even though it's one bird, that whole flock of birds that's producing those eggs have to be cold, and you have to re-establish a new flock of birds and test it. So it could be it could be a year before you're up and running again. The mode of attenuation for MVA, it doesn't replicate in humans. It is still live, it's still causing infection, but it doesn't produce progeny virus. And how that mode of attenuation is not known. During the attenuation, there was a lot of deletions made, and they simulate those deletions in vaccinia virus, but it still replicates. So they're not. So the process of it not being able to produce progeny virus is still quite unknown. Another one that was used, and this is used more as a vector vaccine and quite popular in the veterinary field, is NIVAC, and that was using more of a rational approach for attenuation. So what they've done is they removed a number of host range genes, so that it no longer was able to replicate in mammalian cells, and at a point where it can only replicate in the chicken embryo fibroblast cells. Again, because it doesn't actually replicate that well in humans, it doesn't produce so much of the antigens that you want to express from that vector. So the immune response tends to be weaker, and therefore you need multiple administrations and a high dose as well. So you need to give like 10 to the 8 infectious units, and that's the same for MVA. To get a really good immune response, you need to give 2 lots of 10 to the 8. When you compare that to the smallpox vaccine, which is about 10 to the 5, they're quite large doses, and it means the number of doses per batch starts to get smaller, so the cost of goods and manufacturing start to increase. So we took that on board. So we made our vaccine. So we just removed one essential gene, and that gene is responsible for viral assembly. So it still was infective, it was still live, it still transcribed its genes. The genome still replicates, and in the genome replicating, you get gene amplification. So you go from 1 genome to 100,000 genomes, which also corresponds to 100,000 copies of your target antigen. So the expression levels are still quite high. It can't produce progeny virus upon vaccination, so it's safe, equivalent to MVA and IVAC. And because we haven't adapted to its cell line, it took host range genes, that it still has the capability to infect different cell substrates. And really, for manufacturing, you just need to pick a cell substrate and get that cell substrate to express that missing viral protein. So when you infect that cell substrate, the virus then finishes its life cycle and produce progeny virus that you can harvest. So when we designed our thing, we also looked for best way for, I guess, upscaling efficiently is to have a self-suspension cell substrate that can be grown in stirred tank bioreactors. So that's what we aimed for. Viros and Ceph cells are Hadron cells. They need a cell substrate to grow on, which means that they're surface area demanding. Having something that's a single cell suspension, then your upscaling is more on volume, and volume, can take less of a floor space than culturing, where it relies on surface area. Our vaccine is still highly immunogenic. We can show in a previous version to Simvax SCV, you can stimulate a very good immune response at 10 to 7 in monkeys, and that was done with our Zika virus chikungunya vaccine. The difference between SCV and Simvax is that we found an easier way to genetically engineer them to express multiple antigens. So we've cut that time down from about, if you want to put 4 or 5 antigens into the vector, that can take in almost a year, whereas now we got it down to a couple of months, which again, for what's currently happening now, maybe in the future, that's going to be a big, a strong point there for quickly making vaccines for emergency uses. So on this slide, I've sort of done some comparisons, and at the end of the day, you can have these vaccine solutions that really good, in terms of immunogenicity, what they can do. But if you can't manufacture them to volume, then they got, you know, that's their Achilles heels. So we sort of looked at the other way around. We didn't look to see what, you know, we want to get a fantastic vector or anything. We thought, you know, what's the thing that where we can get hundreds of millions of doses very quickly. And this is why we chose poxviruses. They titer quite high. This is why we chose suspension cultures, so we can grow them in bioreactors. You know, and just to bring that point home, you know, with our cell sorter, we can culture to 10 million cells per mil. The average production rate of virus per cell is 100 infectious units per cell. So that's equivalent to vaccinia virus infecting HeLa cells. So we're up there, still there, even though it doesn't, it's highly attenuated. Losses during downstream processing. You know, the rule of thumb when I was working with Bavarian Nordic, when we did our manufacturing there, you know, the rule of thumb is 50%, and that's what we're seeing. We're working with the likes of Sartorius, and they firmly believe we can get those losses down to 20%. If you look at a dose of 10 to the 7 infectious units, and then the other thing I would like to say is that our substrate can be expanded quite rapidly. So it can go, the expansion rate is 1 to 10. So, you know, if you start with 1 liter, you can go to 10 liters within a couple of days. Virus cells, it's 1 to 5. And I think Ceph cells is about 1 to 5 as well. So expanding from a working cell bank to a 2,000 liter bioreactor, that's 6 expansion steps over a 12 to 18 day period. And if you look at what the yields, we haven't done these yet. I mean, these are just extrapolated from doing process development work in the lab. At a 50 liter scale, we theoretically would be able to produce 2.5 million. But the important thing down there, 2,000 liter scale, we can produce 100 million doses. So that's 100 million doses in one batch run. And that's the sort of thing you need to do now, really for the COVID vaccine. If you're going to have to produce 10 billion doses to vaccinate the world, and you don't want to wait 2 or 3 years to be able to do that. As I mentioned before, MVA and the original and the second generation smallpox vaccines are produced in roller bottles. Also, MVA is manufactured from eggs. So MVA is used as a vector as well. So MVA is being used for a number of infectious diseases as well as solutions for cancer. So if you take a look at the 2,000 liter, you'll need about 66,000 roller bottles. Yeah, and you'll need to buy in 120,000 eggs. So these are heavy logistics. You need a lot of floor space, a lot of storage space, and an army of people to handle that amount of logistics. And the culturing volume, you're up into like the 6,000 liters. Whereas with 2,000 liter bioreactor, you can start with a small bioreactor, with a 1 liter one. Pop in 9 liters on top when the cell gets to density, pop in another whatever. So it's just an incremental stepwise. I think I might have missed the slide. So here, so these are what the bioreactors look like. The 1,000 liter bioreactor doesn't really take much of a floor space. So a reasonable 100 meters squared clean room will easily accommodate that with all the accessory things that have to go with that, and the 2,000 liter, you're not into really a warehouse type arrangement. You're still in a small, a medium-sized clean room. If you're looking at the eggs here and the roller bottles, then yeah, the floor space area is going to be immense, and the incubator rooms that these things need to go to will be immense. So the infrastructure is going to it's going to be quite comprehensive. Having said that, there are new technologies around that can even shrink this down further. And one of the technologies that we're looking at is this fixed bed membrane bioreactor. Now, what this is it's sort of a nylon or plastic membrane, and it's spiraled around tightly. And these consist of fibers that trap cells. And and then the media just is flown through it, and it makes contact, and those cells are growing those quite happily. Universe Cells technology is developing this, and if you look at what they call the 200-meter square one, the amount of cells surface area that can grow the cells is equivalent to a 1,000-liter stirred tank bioreactor, and this is handheld. So it's if you look at the previous slide of a 1,000-liter bioreactor, I mean, that's probably about a height of a human here. That's quite a large equipment, whereas now you're down to something that's really small and can be handheld. And there are other size, the 600 meters squared, that produces equivalent of cells of a 3,000-liter stirred tank reactor. And seeding these things is also quite small. To seed this, you only need to grow a 10-liter stirred tank bioreactor to full capacity, and then use that to seed this. This one is a 50-liter. So it's sort of really shrinking the manufacturing process down to to small scale, but with high yields. This is what we're employing. We're looking at developing a high capacity, small footprint manufacturing process. We've gone with these isolator systems, and you can see in the first one, there's the 200-meter square one, and see how small it fits inside these things. These are about the same size of a biosafety cabinet, and they're in, these things are actually glove box isolators, so they maintain a sterile environment. It separates the operator and the external environment from the product. So inside these things, you can be running at this a Class A clean room specifications, and the operators don't need to be gowned up, and you don't need to have these in a series of different clean rooms. Now, if you're doing aseptic sterile manufacturing, which you have to do with most live viral vaccines, meaning you can't do an end-stage sterile filtration, you do the open manipulations in a Class A clean air environment. That has to be then circled around by a Class B, which then has to be circled around by a Class C. So when you enter, you've got to go from C, B, then to your A. With this system, you don't need that. And as these are, again, small footprinted apparatus, you can easily put this into a 100-meter squared lab. And with the 200-meter squared, you can produce, with our Simvax, about 50 million doses, and with 600-meter squared, it's 150 million doses. So these are real top-end commercial scale things in a very small manufacturing capability. The only problem then, it's easy to do this, and you can produce hundreds of millions of doses, but then the nightmare of fill and finish becomes, and this fill and finish here is really just for clinical trial purposes. That then really is a resource demanding process, and we are partnering with Veta Pharma that specialize in this, and they have fill and finish lines that can do 500,000 doses per day. Still means it takes about a year to do hundreds of millions of doses, but a number of those lines going in parallel, so you can shorten down the time. And then there's the logistics of the glass to produce the amount of vials to bottle this vaccine, and the syringes and needles. So I guess that's the weak link in the process, and still needs, I guess, some clever thinking to overcome that in order to streamline that process and be less resource demanding. Simvax, with our thing, you can use it at different ways for vaccination. Standard injection, syringe and needle, it goes into the arm, intramuscular. With liquid formulation or reconstituted freeze-dried vaccine, one of the things that we're looking at, especially with the COVID vaccine, so I believe, location is important for vaccination. If you can get it into the lungs, I think you can stimulate a far better immune response to control COVID or influenza or RSV than if you do an intramuscular injection. So this is one of the things we're looking with our COVID vaccine, a liquid formulation in the same sort of setup as what people use for flu. This is just a picture of a flu mist vaccine applicator. The other thing that we were also looking at is solid dose implants. So this is where the vaccine is formulated in a little tiny shard. This is the sort of like a sugar glass thing. It's just sugar polymers. It's solid, and this is like an injector, so that just punches it into the skin, and then that then dissolves. And it depends on what your formulation of your shard, whether it dissolves in 5 minutes, 10 minutes, an hour, 1 day, or 2 days. It depends on what your goals are. So these are things that we're looking at. We're trying to move away from needle injections to needleless free applications. Our pipeline, well, one of the reasons why Vaxmed did actually was created was really we were asked to produce a vaccine solution for coronavirus, and this came out from some of the people we're talking to. They're right that if the world is producing vaccines, Australia, New Zealand, and some other countries, they're going to be on the lower list of priorities of getting vaccines. So they're worried about that, and they thought, well, we want to have our own manufacturing capability. And then there's a real concern that CSL sort of moved most of their of our vaccines offshore, and there's very little capability in Australia to produce viral vaccines. So we sort of said, okay, we'll jump into the fray. We'll get funding to help us do that. One of the bigger projects that I was involved in before was a universal influenza vaccine. I think with the pox viruses, you can achieve that. There's a lot of highly conserved antigens influenza that can produce cross-reactive non-neutralizing antibodies that can clear influenza, have been shown to clear influenza infections, and more important, cell-mediated immune responses. Now, if coronavirus hadn't occurred, the biggest thing in the news would be African swine fever. This is an infection. This is the Ebola virus of pigs, and it's really decimating the pork industry at the moment, and it's rampant in Southeast Asia. It was regularly reported by in the news, in newspapers before COVID. Now because of COVID, you don't hear anything about it. It hasn't disappeared. It is still raging, and this one's a difficult one to develop a vaccine for, because if you inactivate the African swine fever, it doesn't give any protection. Protection is now known, it's not antibody mediated, it's cell immune mediated. So you need a good vaccine that can create strong T cells. Fortunately for us, African swine fever is a relative of pox viruses, and the method of protection is exactly the same. Smallpox, method of protection there is cell-based, not antibody-based. One of the things that these vector systems are very good, it stimulates a very strong Th1 biased immune response, which puts us into the RSV vaccine development side of things. RSV vaccine was developed in the 1960s as an inactivated vaccine adjuvanted to alum, and when it was given to a number of kids, it actually caused some of those kids to react really badly upon natural infection, and that was because the vaccine was formulated with alum, which creates a good strong Th2 immune response. Now, if you into allergies is caused by an antigen specific Th2 immune response, and that sort of leads to anaphylaxis. And this is what some of these kids who got vaccinated with the RSV vaccine in the 1960s, they came down with anaphylaxis upon natural infection. So the current thinking or goal today is create an RSV vaccine that has a very good Th1 immune response. And top of the list of doing that are the pox viruses. So this is something that we're also doing. we've been approached by the Cystic Fibrosis Foundation, and they asked, I didn't know this. People who die from cystic fibrosis are actually succumbing to bacterial infections in the lung. These antibiotic resistant bacteria, and that's the end game for them. So creating a vaccine against two of the major ones, which is the Pseudomonas aeruginosa and Staph aureus, is a good thing. People with cystic fibrosis have a heavy course of antibiotics, but sooner and later those bacteria become antibiotic resistant, and there's nothing else that can be done for them. These bacteria have a number of serotypes of like 34 serotypes. So creating like conjugated vaccines that are serotype specific, this is not really going to be a solution. Now, a lot of these bacteria have been shown that the way the body copes with these bacterial infections is they create what's called a Th17 immune response, and it's the innate. This is your first line of defense that clears the bacteria. But in the adaptive immune system, it's the Th17 that controls the innate immune response. And again, pox viruses are very good at creating a Th17 immune response. So you can create an antigen-specific Th17 immune response, then it stimulates an innate immune response that targets those vaccines. And this is an antibody process. It's non-neutralizing antibodies, but it's antibodies that clear bacterial infections. So we're in the process of getting money from the Cure for Cystic Fibrosis Foundation. They like our approach, and we've been promised to get some money to do that. So that's it. That's the end of my talk.
Thanks so much, Paul. That's very interesting, and it's thank you for including all the other vaccine work that's being looked at by your company as well, because it's very easy for us to forget that there's lots of other things happening in the world at the same time as coronavirus that still needs the attention of our brightest minds around the world. We do have a little bit of time for a few questions, and you touched on this in your presentation, but because of the number of questions coming through for this, I just wanted to perhaps get some more comments from you about, once a vaccine is approved, the next challenge is to mass produce it and get it to the ends of the globe. So with each country potentially working on a different type of vaccine, do you see the scale up and the fill and finish process and perhaps the technology transfer to be the next challenge that we're all facing?
Yeah, look, I think each country needs to be able to manufacture their own vaccines, because I mean, if you look at the population of UK, 60 million, and you can't wait a year for somebody to produce that amount or to make sure that the whole population is covered. So if they focus their resources on to that, likewise America, and I think it's the same for Australia. Australia needs to have their own capability. They shouldn't be relying on other countries to supply vaccines for them. And one of the things that when we were talking about developing our vaccine for smallpox vaccine, which everybody keeps asking us, is this something we're doing? I was quite surprised when somebody in Australia we were talking to said that they get their smallpox vaccine from Germany, but it's only a small amount. And he said if that ever gets released, every country will put their walls up and they won't be dishing out their vaccines to other countries, and and Australia will be left vulnerable. So that was one of the things that they were encouraging us to do this, and I think it's the same for influenza. That's the scare as well. And one of the scare was that and it had occurred in the last pandemic when CSL produced a vaccine, but then they gave priority to America over Australia, sort of thing. So it is an expensive process, manufacturing. It is resource demanding. But with modern technology now, you can streamline it, and you don't need an army of people to manufacture. And the way that we're sort of looking at is that we can produce the hundreds of millions of doses. I mean, Australia's less than 30 million, New Zealand's about 5 million. you know, we can then, the idea is that we will then ship bulk vaccine to each of the countries and let them do the fill and finish, you know. And I mean, that's probably the way to go. It's something we could never do. It will take us 10 years to be able to do, you know, set up fill and finish to, you know, to meet, you know, mass global demands, you know. Commercial demands for, like, you know, chikungunya, Zika, RSV, that's fine. We can do that, you know, establish it here. But if you're talking billions of doses, you know, then yeah. And you want it really cheap as well, because it's not just America, Europe, you know, Japan, you know, Southeast Asia and Africa, and they just don't have the monetary resources to buy expensive vaccines. You know?
Thank you. The other theme in some questions coming through are the long-term durability of the immune response, and perhaps you know, I open this question to both of you. Is this a known element of the vaccine development at this stage, or do we have to see how it turns out?
Let me just go first. So again, with live viral vector vaccines, there is longevity immune responses, and we've shown with our chikungunya Zika virus vaccine that, you know, when you vaccinate, this is in my studies at this stage and monkey studies, the antibody titer does slowly go up with time, and we're talking over a 6-month period. And you know, you have long-lasting plasma secreting cells. These are cells that actually make the antibodies, plus the antibodies stay there. Memory T cells are immediately produced. So recall of you know, of T cell responses to antigens is there years after. Now, I'm not saying that, you know, as we've originally thought with the smallpox vaccine, you had lifelong immune response. You do have long-lasting immune responses over, you know, a period of 10 to 20 years. You know, you'd probably boost just to be on the safe side, but you know, if you don't, then you're not in a dangerous position. Yeah, so with ours, we've shown that with just a single shot vaccination, and that's the thing that we're trying to do, is that, you know, if you're going into Africa, and I think somebody mentioned to me that, you know, it's very well with the prime boost with the MVA and the adenovirus, but they're not coming back for their second shots, you know, and you know, they're nomadic, and, you know, to get the vaccine, that's, you never see them. So in those situations, you really do need to have something where you just give one shot and then forget about it, you know. And you know, and we are seeing that with our vaccination in a monkey study with Zika virus and chikungunya. One shot was enough to give total protection.
I mean, I'll just add that I don't think we know for coronaviruses. I mean, we can extrapolate from what's been done in veterinary vaccine development for coronaviruses into some live vaccine work done there. But I mean, realistically, we don't know. I think it seems like there's studies saying that you get long-term immune memory from people who've recovered from SARS. So that's good. We're also seeing a normal decay of neutralizing antibody response from people who were infected earlier in the year. That's normal. There's some anecdotal evidence that some folks are potentially being reinfected, although that hasn't been confirmed. And so I think, you know, and the fact that we don't have a human vaccine for a coronavirus. So I think all of that just goes to say that it'll get answered in time, and, you know, whether a live virus vaccine provides, you know, a better longer term immune response versus, you know, say a subunit or other type of vaccine, we will see. What is clear is that you need a strong and balanced immune response, and that, you know, hopefully we can avoid those side effects that we talked about for RSV and some other animal work that was done in the SARS and MERS area, where we did see enhanced disease with vaccine development. So if we get a balanced immune response, Th1, Th2, and we can avoid enhanced disease, then I think we're in good shape. But jury's out on how long it'll last.
Yeah, there's also been a recent publication. It was in bioRxiv, so it's not peer-reviewed, where they showed that some people who hadn't been infected with SARS did have T cells reactive to SARS antigens, and they believe that because coronaviruses in general causes common colds and stuff, that a previous infection with a common cold might actually set you up for, I guess, a less severe outcome or a asymptomatic thing. But you know, it's not peer reviewed, so you have to take that with a pinch of salt, and that's something, you know, it needs to be studied as people develop these vaccines. I think this is the start. I mean, you're right, Trent. I mean, you know, as we go along, these are things that needs to be studied. You know, once the vaccine gets out there, what are the what are the mechanisms of true immunity and how long it lasts, and you know, and will it set you up for enhanced disease if you get natural infection? You know?
Great. Okay, thank you both. Some other questions that have come through are related to SARS and MERS. So obviously, COVID-19 pandemic is unprecedented for our current generation, but we all very much remember SARS and MERS and the devastation those viruses left. What's happened to those viruses, and have they disappeared or been eradicated? We don't hear that much about them anymore.
MERS is still going around. SARS, yeah, that sort of seemed to disappear, but I don't know, is the current COVID, you know, an evolution out of SARS? You know, I have read somewhere. I was on a webinar with One Health. This is an organization similar to CEPI, and some of them, you know, some thoughts that, you know, SARS-2 actually came from humans, not from animals. It might have been brought in by somebody that visited a market and spread it around. And if it did come from humans, is this something that was a residual thing of, you know, the first SARS? You know, and the RNA viruses, so they mutate really rapidly, and over like the 10 or 15 years, it's quite easy to say there'll be enough mutations to make it look, you know, sort of distinct from SARS-1, but
and I think the important thing to remember is that the surveillance work that's happening with organizations like that clearly show that, you know, whether it's in northern Thailand or southern China, you know, there are lots of people with these potential viruses in their system, and you can do serology and pick up all kinds of things. So there's viruses jumping out of intermediate species into humans all the time, so we don't know. The big difference between SARS and MERS is that it appears they're not that great at transmission, and also they seem to only transmit when people are truly sick. I mean, the big difference with you know, this virus is that you get a large number of people who are potentially seemingly asymptomatic but still shedding virus. And even the people who do get more severe disease seem to be able to shed virus prior to onset of serious symptoms. So, you know, from a transmission point of view, this has got a whole different dynamic. And also because of the big spectrum of disease from absolutely nothing to, you know, death, you know, it sort of found that sweet spot of all the things that, you know, a virus that we would think should cause a pandemic has. And that's the really difficult thing with SARS-CoV-2.
Yeah. It is that thing where shedding before onset of symptoms is the one that's causing the big headache at the moment, because you just can't control it or for it. Because if you don't know if somebody's infected, then you know, there's no way of taking that person out of the system while he's carrying the virus. You know.
Okay, I think we will we'll wrap up today's webinar. I want to thank you both for your time and your informative and I would say for me personally comforting to see that all the work is being done and all the progress that's been made in a very short time. So thank you again for your presentations, and I wish you both and your teams a speedy progress on your projects, not just for coronavirus, but for everything else you're working on. And I'd like to thank the RACI Pharmaceutical Sciences Committee for putting together this webinar and hosting it for everyone today. You'll be able to find a recording of this webinar on the RACI website very shortly. And thank you all for joining us.
You're welcome. Thank you for the invitation to speak.
Pleasure. All right. Thank you. Bye.
Thank you. Bye.
Bye.